Okay, so welcome to this next video in which we are discussing the use of phosphodiesterase inhibitors to treat asthma. In this next video, what we're going to start talking about is the actual phosphodiesterases and how phosphodiesterase inhibitors are going to be useful in the prophylactic treatment of asthma. Okay, so let's firstly discuss phosphodiesterase enzymes. It's a big long name for a whole class of enzymes. Okay, so phosphodiesterase enzymes are also occasionally called, their full name is cyclic nucleotide phosphodiesterases. Right, and I'll just add a little S there. Right, okay, so let's start off with talking about what phosphodiesterases are actually going to do, and then we'll see all of the different families of phosphodiestrases. Okay, so phosphodiestrases are going to break down the two main cyclic nucleotides, which are cyclic AMP, cyclic adenosine monophosphate, and also cyclic GMP, which is cyclic guanosine monophosphate. So let me just draw the structure of cyclic AMP and cyclic GMP. Okay, so I'll draw a little cartoon structure for both of them. So, firstly, their names are cyclic adenosine monophosphate. So let me write its name out in full. Okay, so it's cyclic adenosine, adenosine, or in the case of cyclic GMP, guanosine, so I'll put a / guanosine, so fill in which one you're talking about. So for this one it's adenosine, for this one it's guanosine, and then the final bit is the same, monophosphate. So the first and final bits are the same, it's the bit in the middle which varies between cyclic AMP and cyclic GMP. Right, okay, so let's now show the structure of this. So firstly we need to discuss what's the structure of adenosine and guanosine. Okay, so adenosine means the organic base adenine, so I'll just draw that as a rectangle with an A in it, okay, bound to a ribose sugar, which is like this cycle here, this pentameric structure, where you have 4 carbons and then 1 oxygen up here, and then a 5th carbon coming off up here. So this is the structure of adenosine. The structure of guanosine is then a ribose sugar, so here's the ribose sugar, where you have a guanine organic base coming off the ribose sugar here. So this is the guanine. Right, now we need to deal with the cyclic adenosine / guanosine monophosphate. So if we wanted to create adenosine or guanosine monophosphate, all we do is stick a phosphate group coming off this 5th carbon here. However, we want cyclic adenosine or guanosine monophosphate, so the phosphate group isn't just going to be attached to this 5th carbon up here, it's also going to be attached to the 3rd carbon. Okay, so you form this extra ring basically here, this cycle, and this is the cyclic in cyclic AMP or cyclic GMP. So here is the phosphate group that's linked both to the 5th carbon and the 3rd carbon of the ribose sugar. Okay, now I'll also draw the ribose sugar in blue. Okay, and I'll colour in finally the organic base, which is either adenine or guanine, in orange. Okay, so phosphodiesterase enzymes work by cutting the bond between the phosphate group and the 3rd carbon. Okay, so what they yield are just adenosine or guanosine monophosphate. So let me show the structure here. So I'll only draw one of them, okay, and you can put in the appropriate organic base. In fact, maybe I could put adenine / guanine here, okay, so A/G there, okay, and now you've got the 5th carbon just with a single phosphate group up there, and you no longer have the attachment of the phosphate group to the 3rd carbon of the ribose sugar, okay, so you no longer have that second cycle in the structure of the cyclic nucleotide. Okay, so this now is just adenosine or guanosine monophosphate. So it's either AMP for adenosine monophosphate or GMP for guanosine monophosphate. Okay, and I just want to emphasize that the sugar that you have there is ribose, not deoxyribose. It's not the sugar that you would have in DNA, it's the sugar that you would have in RNA, ribonucleic acid. So this is ribose. Right, okay, so that's what phosphodiesterases are going to do. They're going to cut this bond here in cyclic AMP or cyclic GMP to break them down to AMP or GMP, so to take away the cyclic. Right, so what different enzymes exist? Because there is a huge number of enzymes that exist. So let's try and get some scope on this complexity. Right, so there are many different types of phosphodiesterase enzymes, okay? And they are mainly made up of dimers of proteins. So firstly, we will start off with the different proteins which you can use to assemble phosphodiesterases, and then I'll talk about how you can actually assemble an active enzyme. So, firstly, let's start off with the families of phosphodiesterase proteins. So to actually make a functional enzyme, you're going to have to assemble these into bigger structures, which hopefully I will make clear in a moment. Okay, so by the way, PDE is often used for phospho is the P, di is the D there, and then esterase is the E, so phosphodiesterases. Okay, right, so there are 11 different families into which the genes which are used to make phosphodiesterase enzymes are categorized, basically. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Okay, so 11 different families. These are not individual genes, these are families, so they're going to have genes within them. Okay, so there is the phosphodiesterase 1 family, so you would put PDE in front. Then there is the phosphodiesterase 2 family, and it goes on all the way down to the phosphodiesterase 11 family. Okay, so this is how you categorize the genes, basically. Now, within each family, there are multiple genes, okay? Now, in some families, there's only one gene, but some families have multiple genes, okay? And they're categorized into a family depending on their structure, basically. They all have a very similar active site, okay? They all have a very similar catalytic site, but they can vary in the other bits that surround the catalytic site. Okay, so for instance, in the first family, there are actually 3 separate genes, okay, labeled phosphodiesterase 1A, okay, and then it goes on, phosphodiesterase 1B, and then finally phosphodiesterase 1C. Okay, in the second family, there is only one gene, and this is called phosphodiesterase 2A. Okay, so this codes for a protein that would then be called phosphodiesterase 2A protein. Okay, in the third phosphodiesterase family, there are 2 genes which will be called phosphodiesterase 3A and phosphodiesterase 3B. Okay, again, both of these code for proteins which would then be called phosphodiesterase 3A protein and phosphodiesterase 3B protein. Okay, in the fourth family, there are then actually 4 genes, okay, and the fourth family is going to be extremely important in asthma, okay. So there is phosphodiesterase 4A, there is phosphodiesterase 4B, there is phosphodiesterase 4C, okay, and then finally there is phosphodiesterase 4D. Okay, so if there's one family to memorize, it's this one, family 4. Okay, family 5 then. In family 5, there's only one gene, okay, and this is phosphodiesterase 5A. Okay, and that will make phosphodiesterase 5 enzymes, which are famously the target for the drug sildenafil, more commonly known as Viagra. Okay, in the 6th family, there are 3 genes. There is phosphodiesterase 3A, phosphodiesterase 3B. Sorry, what am I doing? 3? Sorry, there is phosphodiesterase 6A. So turn these into 6s somehow. Okay, there's a 6. 6B, oh dear, it was going so well. Phosphodiesterase 6C. Okay, so there's phosphodiesterase 6A. I'll have to put something there to make that more obvious. Like that. 6A. Okay, phosphodiesterase 6B and phosphodiesterase 6C. And these are all going to be very important in the rod and cone cells in the retina. Okay, so they're important in phototransduction. Okay, right. Well, the signaling cascade that follows phototransduction, they're not actually involved in the phototransduction, but they're involved in the signaling cascade that follows phototransduction. Okay, in the 7th family, there are then 2 genes, okay, and these are known as phosphodiesterase 7A and phosphodiesterase 7B, okay. In the 8th family, there are then again 2 genes, okay, known as phosphodiesterase 8A and again phosphodiesterase 8B, okay, and then finally for 9, 10 and 11 there are 1 gene each, known as phosphodiesterase 9A, phosphodiesterase 10A, okay, and then finally phosphodiesterase 11A. Okay, so overall there are 21 genes. I think that's right. Let's count them up. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21. Yes, so 21 known genes in humans. Okay, so if you were being really pedantic, you would stick in front of the names of all of these genes a little HS. Okay, so for instance, to take 11A as an example, you would call it HS phosphodiesterase 11A, which means Homo sapiens phosphodiesterase 11A, so it refers to our species, because in other species this is quite different. But of course, we're only interested in humans. Right, so we have these 21 genes. These all code for proteins. Now, are these proteins themselves functional enzymes? Well, the answer is no, unfortunately. It gets a little bit more complicated still. Okay, so you're going to basically assemble them into larger structures, which are then going to be functional enzymes. Okay, so usually it's very simple. There are a few exceptions. Okay, so there is a very nice rule for how you assemble them. Okay, so the very nice rule is that generally all you have to do is take 2 proteins, 2 phosphodiesterase proteins, stick them together, and that will make you the phosphodiesterase enzyme. And it's even more simple. You take identical proteins, so this is usually a homodimer. Okay, so what you would do is you would pick your gene, okay, you'd make that protein twice, and then you'd stick those 2 proteins, the 2 copies of the identical protein, together to make a homodimer, and this is then a functional phosphodiesterase enzyme. Now, this is true for all the families apart from 2 that are awkward. Okay, so the 6th family here is awkward, so I'll highlight it, and also the 1st family is awkward. So if you're talking about any other genes apart from the 3 in family 6 and the 3 in family 1, you assemble homodimers like this. So pick any of the genes that aren't in family 1 or 6, take that gene, copy it make 2 mRNAs from that gene, translate both of those mRNAs into proteins, stick the 2 of them together, and you've got a functional phosphodiesterase enzyme. And that phosphodiesterase enzyme that you've got would then be named after the gene which makes both of the subunits of the enzyme. Okay, so for instance, if I took the phosphodiesterase 4A gene and made 2 copies of that protein, stuck them together, then the enzyme I would get would be called a phosphodiesterase 4A enzyme. Okay, so it works nice and simple for everything apart from the first and the sixth family. Okay, so I'll talk about the sixth family in a bit of detail, and then I'll just sort of brush over the first family and just say that it's complicated. Okay, so the sixth family is nicely studied because it's so important in the eye. Okay, so you have two main forms of phosphodiesterase 6 enzyme. One that is in rod cells, okay, rod cells, and the other which is in the cone cells. Okay, so you have two different types of phosphodiesterase 6 enzyme in the rod cells and the cone cells. And the functional enzyme isn't this time a dimer, but a tetramer. Okay, so you have to put 4 proteins together to make a functional phosphodiesterase enzyme. Okay, now, basically the one in rod cells is made up of A, B, and then 2 C's. So you put you put one phosphodiesterase 6A here, you then put one phosphodiesterase 6B here, you then use phosphodiesterase 6C twice to make 2 proteins and put them here. That then forms the phosphodiesterase that is found in rod cells, and it's just generally called a phosphodiesterase 6. Okay, and then in cone cells you have a slightly different one. In cone cells, you don't use the phosphodiesterase 6B gene at all. You just use 2 A's with 2 C's. So you use the phosphodiesterase 6A gene twice, make 2 copies of that protein, stick them in here, and then use the phosphodiesterase 6C gene twice, make 2 copies of that protein and stick them in here, and that will make you the tetramer that is in cone cells. Okay, so these are phosphodiesterase 6 tetramers. Okay, now for one it's very, very complicated. Again, they form tetramers rather than dimers. But for all the other families, and for the family that we're particularly interested in, which is this 4th family here, they're going to be in bronchial smooth muscle cells, and also mast cells is going to be very important. Those form dimers, and they form homodimers, which is just lovely. Okay, right, so let's now talk about the selectivities of the different phosphodiesterase enzymes. Okay, so different phosphodiesterase enzymes have different selectivities. So remember, we talked about how phosphodiesterase enzymes they broke down both of the cyclic nucleotides. Now this isn't quite true. Not all phosphodiesterases will break down both cyclic AMP and cyclic GMP. Some will break down both of them, but some will specialize in breaking down cyclic AMP, and some will specialize in breaking down cyclic GMP. So what are these specificities, basically? Okay, so let's start off with those that will break down both. Okay, so the ones which will break down both we will highlight in orange here. So 1, 2, and 3 all break down both. So all phosphodiesterases that are within the first family, all the phosphodiesterases that are constructed from proteins in the first family, they will break down both. Okay, both cyclic AMP and cyclic GMP. All the phosphodiestrases that you can produce from the second family, which is obviously just the homodimer of 2 phosphodiesterase 2A enzymes, proteins, these will break down both cyclic AMP and cyclic GMP, and then the same for the third family. Now it's also true for these final 2 down here, 10 and 11. All of the phosphodiesterases that you construct from proteins in the phosphodiesterase 10 family, and also all of the phosphodiesterases that you produce from the phosphodiesterase 11 family, they break down both as well. So orange is both cyclic AMP and cyclic GMP. Okay, so these are both cyclic AMP and cyclic cyclic AMP and cyclic GMP. Right. Now let's talk about the ones that are selective for cyclic AMP. Okay, so the ones that are selective for cyclic AMP are 4, 7, and 8. So 4, the star of the show here, is selective for cyclic AMP, and 7 and 8 are selective for cyclic AMP. So these are the ones which will only break down, won't only, but much prefer to break down cyclic AMP than cyclic GMP. Okay, so all of the phosphodiesterase enzymes which you create from the 4th family, which of course are these 4 different types here, because you'll make the phosphodiesterase 4A homodimer, you'll make the phosphodiesterase 4B homodimer, you'll make the phosphodiesterase 4C homodimer, and the phosphodiesterase 4D homodimer, and the same for 7 and 8. Okay, all of those enzymes are going to be selective for cyclic AMP. Now finally, which I'll highlight in turquoise, the remainders 5, 6 and 9 here, these are selective for cyclic GMP. So 5 and 6, which we know are very physiologically important. 5 is the target for Viagra, 6 is very important in the retina, and 9 over here, those are all selective for cyclic GMP. So this one, and this family down here, they are all selective for cyclic GMP. Right, okay, so now let's talk about phosphodiesterase inhibitors, drugs which can inhibit phosphodiesterase enzymes. Now, we're going to discuss firstly the xanthine alkaloids, because I don't think we can really discuss phosphodiesterase inhibitors without actually discussing the xanthine alkaloids. Now, these are non-selective inhibitors. They inhibit all phosphodiesterase enzymes pretty much. Okay, so let's discuss the xanthine alkaloids. Okay, right. So, the first one that's very, very important, okay, is a drug known as theophylline. Okay, and this is going to be useful in the treatment of asthma, and it is used in the treatment of asthma. Oral theophylline is used in the treatment of asthma. Okay, now we have better drugs than this now, because the problem with theophylline is that it inhibits pretty much all of these phosphodiesterase enzymes. Now, we needed to find which phosphodiesterase enzymes were actually important in helping asthma, basically. We know that this drug is good for asthma. It's good, it works to treat asthma. The problem is it's blocking all the other phosphodiesterase enzymes all over the body, okay, and that will lead to side effects. So if we could find out which phosphodiesterase enzyme was particularly important for theophyline's action against asthma, then we could get drugs that were selective for that specific phosphodiesterase, and then hopefully they'd have fewer side effects. Okay, but we'll talk about that in a moment. So theophylline is a selective xanthine alkaloid. Is a non-selective inhibitor of all phosphodiesterase enzymes, and it's a xanthine alkaloid. Now there are other xanthine alkaloids that also are non-selective inhibitors of all phosphodiesterases. Caffeine is an example. Another example is theobromine, which is a molecule that's found in chocolate. Okay, so both caffeine and theobromine are non-selective inhibitors of phosphodiesterase enzymes as well. Now they're not used to treat asthma, but theophylline is. So I'm just mentioning those for completeness. Okay, right. So now let's talk about why inhibiting these phosphodiesterases is helpful in asthma. Okay, so basically, in your bronchial smooth muscle cells and also in your mast cells, okay? So in mast cells and bronchial smooth muscle cells, so I'll put these down here. So in mast cells and also in the bronchial smooth muscle cells, you have phosphodiesterase 4 enzymes, okay? So bronchial smooth muscle cells. The major type of phosphodiesterase that is expressed in bronchial smooth muscle cells and mast cells is phosphodiesterase 4. Okay, and now we'll just abbreviate it to phosphodiesterase 4, PDE4, and we won't distinguish between the 4 different types of phosphodiesterase 4. We of course know that there are 4 different types of phosphodiesterase 4 enzymes. There's the phosphodiesterase 4A homodimer, there's the phosphodiesterase 4B homodimer, there's the phosphodiesterase 4C homodimer, and then finally there is the phosphodiesterase 4D homodimer. And it even gets more complicated than that, because there are actually different splice variants of these genes, which I didn't tell you about. So even if you're coding, even if you're coding with a single one of these genes, you can actually make different proteins from the same gene by having different splice variants, i.e. using different exons to actually make the protein. Okay, so we'll just abbreviate this mess to PDE4, and we'll know that that means a phosphodiesterase enzyme that is produced by one of these, by these genes, which are all put together as being similar enough and producing similar enough proteins that they deserve to be put together into a family. Okay, so we'll just refer to a phosphodiesterase 4 enzyme from now on. Okay, now what does this enzyme do? Well, we've seen that phosphodiesterase 4 enzymes are selective for breaking down cyclic AMP, so it's breaking down cyclic AMP. Right, so theophylline is useful in treating asthma. It's useful for preventing asthma attacks. So it must be going in and blocking the PDE4 in the mast cells and the bronchial smooth muscle cells. So what would happen if we blocked PDE4 in mast cells and bronchial smooth muscle cells? Well, you'd stop breaking down cyclic AMP. So cyclic AMP level within the cytoplasm would go up, basically. If we reduced the activity of phosphodiesterase 4, then we're no longer breaking down cyclic AMP, so cyclic AMP would accumulate in the cytoplasm of our mast cell and our bronchial smooth muscle cell. Now, this maintains relaxation of bronchial smooth muscle cells, so it keeps bronchial smooth muscle cells relaxed, so it's more difficult to make bronchial smooth muscle cells contract, and I'll explain exactly how in a moment. Okay, so it's going to keep bronchial smooth muscle cells nicely relaxed. It's also going to actually stop mast cells from secreting their pro-inflammatory mediators. So it stops mediator release. Okay, so remember, in allergic asthma, what happens is you breathe in the allergen, okay, and this causes the activation of the mast cells, and they release those 4 inflammatory mediators, which then lead to the asthmatic attack. So, if we inhibit the phosphodiesterase 4 enzyme within mast cells, cyclic AMP is going to go up, and this is going to stop the mediator release within mast cells. So that's one of the ways it's going to help prophylactically treat asthma. In addition, you're also going to be keeping the bronchial smooth muscle cells relaxed. So even if some mediator does get released, and of course you're not going to block it all, you're going to dampen it down, then the effect on the bronchial smooth muscle cells is going to be hugely reduced. So this is going to lessen the severity of the asthmatic attack hugely, and maybe it will make it subclinical, basically. Okay, so this is the basis of the use of theophylline to treat asthma. Okay, so if we could design drugs that selectively block phosphodiesterase 4 enzymes, then this would be a very good thing, because it would have all the positive effects of theophylline as far as treating asthma is concerned, and it wouldn't have nearly as many side effects as theophylline because it wouldn't inhibit all the other forms of phosphodiesterase enzymes. So there are many drugs which inhibit phosphodiesterase 4 enzymes and selectively inhibit phosphodiesterase 4 enzymes. Now, the one that is currently used prophylactically for asthma and is taken orally is Rolipram. Okay, so Rolipram is a selective phosphodiesterase 4 enzyme inhibitor, and it is currently used to treat asthma. Okay, now some other drugs that are currently in development are roflumilast. Okay, this is also a selective phosphodiesterase 4 inhibitor, so roflumilast, and also silomilast. Okay, silomilast. Right, now again, there are finally some drugs that don't have proper names yet, which means that they certainly aren't used clinically, and they're not even in clinical trials yet. Okay, so another drug which is used to treat asthma is YM, okay, 976. And then finally also you have a drug known as SB 207 499. Okay, 207499. So SB207499 and YM976, those are both also drugs which are selective inhibitors of phosphodiesterase 4. So here are some drugs which selectively inhibit phosphodiesterase 4 enzymes. So Rolopram is in current clinical use. As far as I'm aware, roflumilast and solomilast are in clinical trials to be used for the treatment of asthma. And then there are also some experimental drugs here which are YM976 and SB207499, which are also selective phosphodiesterase 4 inhibitors, and potentially might have a use in the future. Okay, so these drugs will also block phosphodiesterase 4 enzymes within mast cells and bronchial smooth muscle cells, and therefore cyclic AMP will accumulate in the cytoplasm of these cells. That will cause the bronchial smooth muscle cells to remain relaxed, and also it will reduce the mediator release from mast cells when the mast cell is activated by the allergen binding to the IgE molecule that is docked on the Fc-epsilon-R1 receptor. Okay, right, so we'll call it there for this video, and in the next video what we'll do is discuss the mechanism by which raising cyclic AMP levels within smooth muscle cells is going to maintain relaxation of the smooth muscle cells and prevent them from contracting.